Now, applications of DNA nanostructures in biological applications. For starters, we can make a cellular signaling path with the help of DNA nanostructures. Signaling using transforming growth factor (TGF) is seen to be dependent on clustering of the receptor particles. By functionalizing the pattern, a DNA nanostructure with closely spaced peptide molecules which bind to TGF receptor, sensitivity of these cultured cells towards TGF was marginally increased. The authors also proposed and partially constructed a multivalent thrombolytic delivery system to measure and deliver human tissue plasminogen activator and plasminogen, two important molecules in thrombolytic pathway. Now, construction of semiconductor nanocrystal arrays of quantum dots using DNA. Many studies on conjugation of semiconductor nanocrystals with biomolecules have been done. These nano-sized conjugates are biocompatible and serve some advantages other than organic dyes and lanthanide probes. The emission wavelength of quantum dot nanocrystals can be regularly tuned by changing the dot size, and a common light source can be used for simultaneous excitation of all different sized dots. The perfect optical properties make these dots ideal for europhoresis for multicolor and ultra-sensitive applications in nanobiotechnology and nano-level bioengineering. Now, DNA nanostructures and theranostic applications. We have already seen the unmatched property of self-assembly of DNA nanostructures and also nanostructures in different dimensions. Since biological properties of the DNA can lead to use of it in other fields of biology like therapy and diagnostic, it can be used as biosensor for such applications. For example, DNA tetrahedra, which is 3D structure which is rigid and has well-defined structures. When it is created with various probes, it provides different types of structures for biosensing. Such structures also improve target accessibility and sensitivity. Other properties like AT and GC pairing and structural features of nucleic acid, where single-strand DNA provides flexibility and double-strand DNA provides rigidity, makes it easier to create structures. Nowadays, technology like DNA origami also provides great help in creating structures. Now, illustration of DNA biosensors. Using the famous thiol gold chemistry and DNA hybridization on the gold surface, eDNA sensor was created. However, there were lots of factors to be controlled to make it work properly, like interstrand entanglement and electrostatic repulsion between strands. This was created by using property of structural switching of 2D DNA stem loop structure. There would be one target DNA to which our complementary 2D DNA hairpin structure will bind and will produce the electric charge, which will get measured with the help of the transducer. Just by changing the probe of the eDNA sensor, property of whole sensor can be modified. For example, ATP sensor was created just by using the structural switching of anti-ATP aptamer. Now, the delivery system using DNA nanostructures. It was observed that this tetrahedral nanostructures were non-toxic and more stable than linear DNA inside the cell. These tetrahedra were also capable of crossing cell membrane. By using this property, new type of drug delivery system of CpG oligonucleotides was created. In the research they created tetrahedral DNA nanostructures for the delivery of siRNAs to cancer cell of mice. Efforts were also made to create efficient drug delivery system using logical functions. Research by Douglas gives the description of robotic DNA device which can sense the receptor. It contained the barrel-like container locked by two aptamers. They made the use of AND gate such that both the aptamers reaches the receptor, then only the barrel will release the cargo molecules. Such intelligent delivery system can lead biology to better levels.